Accurate cell counting is essential for cell culture experiments. In this segment, we will demonstrate manual counting using a hemocytometer. Before counting, ensure that the cell suspension is homogeneous. Gently pipette the culture up and down to break up cell clumps. If necessary, filter the suspension to remove large aggregates. To distinguish live cells from dead cells, we use trypan blue staining. Live cells exclude the dye, appearing clear, while dead cells take up the stain and appear blue. In a microtube, add 10 microliters of trypan blue. Then, add 10 microliter of the cell suspension and mix gently. Load 10 microliter of stained cell suspension into the chamber of the hemocytometer, allowing capillary action to evenly distribute the sample. Using a microscope, count the cells in the 4 large corner squares. It is essential to count in a consistent manner. Always count the cells touching the top and left borders of the squares to avoid double counting. Automated cell counters streamline the counting process, reducing variability. Simply load the stained sample into the device, and it will provide total cell concentration and viability in seconds. Now, let's review the key concepts and essential calculations for accurately interpreting cell count results. Accurate cell counting ensures reproducibility in experiments. Manual counting is cost-effective but prone to user error. Automated counters provide speed and consistency but require calibration. Hemocytometers work best for general cell culture, while flow cytometry or image-based counting is preferred for high-throughput applications. You can calculate your cell concentration using the following formula. So, for example, if you diluted your sample 1:1 with trypan blue, dilution factor is 2 in this case, and you counted 325 cells in the 4 corner squares. Number of squares counted is 4. This gives us the following calculation. If you want to know how many cells you have in your original sample, just multiply the cell concentration by the total sample volume. For example, if your original sample volume is 5 milliliters, this gives us the following calculation. To assess the health of your cell culture, we calculate cell viability using trypan blue staining. Viable cells exclude the dye and remain clear, while non-viable cells absorb the dye and appear blue. After staining, count both viable and non-viable cells using a hemocytometer. Then, use the following formula to determine cell viability. For example, if you count 150 viable cells and 50 non-viable cells, the total number of cells is 200. This gives us the following calculation. This means that 75% of the cells in your sample are viable. Accurate cell counts are crucial for seeding cells in multi-well plates for assays like MTT or flow cytometry. Knowing the exact number of cells allows for better experimental consistency. In drug screening, knowing the cell concentration ensures that the right number of cells is exposed to compounds, leading to reliable assay results. Correct counting ensures that cells are split at the appropriate density, optimizing their growth and maintaining consistency between passages. Now that we have covered the theoretical aspects of cell counting, let's go over some common mistakes and how to avoid them to ensure accurate and reliable results. If cells are not evenly suspended before loading, counting will be inaccurate. Always mix thoroughly before sampling. Too much or too little liquid in the hemocytometer's chamber can cause inaccurate readings. Load just enough to allow capillary action to fill the chamber evenly. Be consistent with inclusion criteria. Count cells touching the top and left, but not the right and bottom, to avoid overestimation.